continue our learning. So last week we learned about the gene bank data source. There are so many gene bank data source outside, but the largest one in CBI gene bank. Yeah, you have to be familiar with this website. There's so many information here. Spend your time, enough time on here. Also, there is a INE, nucleo AB, okay, European Nucleotide Archive. Yeah, so this is much more in US and European. So to download the data manually, you have to follow how to access here. Like they have some tools like Entrust Nucleotide for gene bank, and also search and align gene bank sequence using BLAST. BLAST is basic local alignment search tool. There's also some other utility. It's quite confusing. And if you go here, it's the same. There's another tools for downloading, etc. So to make sure that you can have productive way of interacting with data, you need to have library. So today I will explain about Python library called Biopython. Okay. So this is commonly used in biology. Please open it. So Biopython is free. It's a Python library. So you can use in Google Colab. Just open your Google Colab and then pip install Biopython. Okay, so let's see some of the features from the documentation. Let's go through for some minutes. And after that, let's get our first try on Biopython using Google Colab. Okay, so it provides you a book, large book, 445 pages, Biopython tutorial and cookbook. I think you don't have to read it all, but let's see few chapters. Later you can get the notebook from this book. I will give you the link. Okay, and some previous workshop on Biopython. So please take these links for you to practice. And let's see what is Biopython. Okay, so it's a library. Okay, collection of library, free and widely used to interact with many file formats of genomic. BLAST, FASTA, yeah. Maybe you never heard about FASTA or BLAST. There is an international standard for that. You have to understand what data you download, what format they have. And Biopython provide parser, means that it can load the file into memory, and then you can interact with the file and information inside the file. So what you can find in Biopython package is ability to parse the file, okay, with many formats, okay. Let's say you are working with GenBank and CBI, and then you download FASTA, okay, or GenBank or PubMed. You can work with Biopython to make sure that you can parse the file and then continue the computation using other Python library or other workflow. Okay. There is a workflow engine for bioinformatics that using Python called SnakeMake. SnakeMake, yeah. Later we will learn about this, but let's focus on Biopython library first this week, so you can have productive way on interacting with the data. Okay, let me continue. So you can also do some code to deal with popular online destination like NCBI, XPC, and you can interface to common bioinformatic programs like standalone blast from NCBI, Cluster W, and Emboss. Yeah. So these tools is like tools for interacting and manipulating the data, but not including the machine learning or any other deep learning library. This tool is focused on data. However, it also provides you some classification data like Carnier's neighbor classification, knife base, and SVM. So some sample of library that you can utilize for classification. You can also have code for dealing with alignment, and then many other features here. Okay, so to install, just run on your Biopython, on your Colab, in pip install Biopython, and then let's try some tutorial from this website. Okay, let me try this one. Okay, so from Tiagoan Tao, okay, this is actually the books, but they make it as a Python notebook. Instead of this PDF, it gives you the Python notebook here. Let's see the introduction. Okay, so this is actually from the cookbook, from the PDF, but you can try some codes that it gives you here. Let's say you want to check the features. This is 1.8, okay? Let's see the quick start. So this is just sample code to work with sequence. So if I copy the code and put here from bio.seq sequence, this means maybe sequence, import sequence and my sequence, and then you can put this literal sequence, okay? So it can contain sequence as a variable here, and you can print your sequence. Okay, and then you can complement the sequence with some operation like complement and reverse complement. Wait. Okay, and reverse complement. Okay, so spend your time in this tutorial for basic, but make sure that also you can use ChatGPT to memorize instead of you have to memorize the code or the library. Yeah. So it's the best way to learn this kind of library is to familiarize with the features and then to automate code generation using ChatGPT. Okay. So first you have to copy paste and look, oh, this is my sequence, and then they print it, and then they count how many G inside the sequence. Yeah, this code is based on Python, so easy to understand. So from this sequence, I just print the length of this sequence, how many digit, 32, and then count number of G is 9, and then print it, okay? So many tutorial here, we don't have time to complete it, but I make it as your homework. Okay, so make sure you understand the capability, and you can use ChatGPT to to ask sample code. Yeah. So let me try. So I will say make synthetic sequence data set. Okay, write Python codes using Biopython. Okay, Python to perform similarity similarity uh, check on sequence data. I hope it can learn. Okay. So let's see. First, install by Python, and then it generates synthetic data because I don't have real data. I just check. Let's see this code. So this is to generate random ICTG. And this is a function to learn similarity between two sequences. Okay. As you can see here, it has bio pairwise to format alignment, means there is a library provided by Biopython to do this alignment, and then you can run the code. Oops. Okay. Sometimes there's an error. It can be because of the version, or let's see. Fix the code. Okay, let's try again. So I will check. Yes, it ran. Okay. So before you will be able to know what is possible inside the library, you have to learn from the cookbook tutorial. Once you learn and you know the facility inside, you can prompt like what I did. Yeah. So you have to finish all of this as your homework. And then you have to make sure that you can do prompting for you not to memorize the line by line of the code. So every time you finish one point tutorial here, you try to do the prompt. Okay. So if you take data mining course today, we talk about cluster analysis, and Biopython provide BioCluster library. Okay, it's based on a paper or C clustering library by Dihun et al. It's based on hierarchical clustering technique, k-means clustering technique, and principal component analysis. Okay, so this is the as after you read, yeah, okay, you know the capability, and then you try to prompt. So let's try to prompt. I will ask, okay, the chat GPT, synthetic genome sequence dataset, write Python codes using to perform clustering, hierarchy, k-means clustering. Okay, I asked him to create any data and then try to do clustering just to see the capability of the library. Okay, so let's try. This is the code. It creates random sequence, this function, calculate nucleotide frequency, and then generate synthetic sequence, convert into vector, perform k-means clustering. Okay, done. And you can also ask if it can visualize the cluster. Revise the code to add visualization of the cluster. It use scatter plot to visualize. So let's check the code, run the code. Yep, this one. So it's very interesting library because it provides you facility to work with sequence as a data. And you can also use library like scikit-learn to perform machine learning, supervised, unsupervised data pre-processing, visualization using Biopython. So please make sure you practice this lab, hands-on lab, yeah, from this. Download the books, yeah. I already put in our folder in Google Drive, and make sure you can generate code after you read the books. Okay, this can help you to download code from NCBI. If you forget the code, you can always come back to ChatGPT. For example, like write sample code to download pasta data from NCBI, and load the data using Biopython. You have to fulfill your email address that's registered in NCBI, and then it gives you the download FASTA. You have to understand the accession number. That why I said you have to read this NCBI beforehand. And then once you get the accession number that you want to download, you can use this code, yeah, to download the data. Once the data is downloaded, then you can process many things using Biopython by this example. Okay, that's it from me. I think after this, you have to continue learn about Terra Bio from Fadin and Cinta. And after we are done with Terra Bio, we will move to Galaxy or SnakeMake or Nextflow. Thank you. Any question? Is clear enough? Okay, so make this, I give you the chat room, this to GitHub as your practice or exercise. Make sure you exercise well. All of this tutorial from the Biopython to make sure that you can productively get data from NCBI or European Nucleotide Archive. Thank you. Please continue with Terra Bio.
Okay, thank you, Mr. Risman. Okay, good morning, good afternoon, everybody. Okay, now in this session, we will discuss about format sequence. Let me share screen first. Okay, I hope my screen already popped up on yours. Okay, I will share the link on chat column. So please open the link. We'll talk about format conversion. So as we know, we need to have PCF file to analyze the gene sequence. So we want to know how to convert from one file to another file. Okay, hopefully all you guys open the link and log in, and let's start. Here I will deliver the materials with Jacqueline and Rasidan. Say hello, Jacqueline and Rasidan, please.
Hi everyone, nice to meet you all.
All right. So the first part will be delivered by Jacqueline. So please, Jacqueline.
All right, thank you, Kashinta. I will be sharing my screen. Right, before we start with the Terra workflow, we'll be taking an overview of the bioinformatics pipeline beforehand. So just to make sure everyone's on the same page and we know what we're going to do with the Terra. So the first step of this pipeline will be the DNA sample. Um, I forgot to mention that this pipeline will illustrate the process of analyzing DNA samples using bioinformatics tools. Here, the bioinformatics tools that we'll be using is Terra. So first we start with the DNA sample. Then it goes through a sequencer. So this process is called sequencing, where the DNA sample is processed by the sequencer to produce FASTQ data. Once it's in the FASTQ format, it will go through an aligner or a mapper, where the FASTQ data is aligned and mapped using the human genome reference models. And then it will create a BAM file or a binary alignment map file. After this, there's an optional step of refinement before going to the next stage, which is variant calling. Here, the BAM file is used for variant calling, and it results in a VCF file or a variant call format file. After it's in a VCF format, the VCFs are analyzed against single nucleotide polymorphism databases and peer-reviewed research. So this is the variant analysis. And from here, a report will be resulted. And this will be all the information compiled into a detailed report, which will then be used for clinical diagnosis or genealogy studies. So this is the overview of the workflow we'll be doing, the pipeline. So then we will move to TerraBio itself. Here I am going to open side by side. You guys can do whichever style you like. I'll do on the left, I'll be opening the tutorial, and on the right, I'll be doing the workflow. If you guys haven't opened the link, you will be seeing a sequence format conversion copy workspace. And in TerraBio, there's a limitation, I believe, where you're only allowed 2 or 3 workspaces. So if you're having trouble opening the link, it may be that you have too much workspaces and you might need to delete one of your workspaces to combat the limitation. So anyways, we'll go back to the workspace. Here, I'll go back to the sequence format conversion workspace. And the first thing we'll be going through is the interleave FASTQ to paired FASTQ. Oh, sorry. Is everyone on this page? If there's any trouble, maybe you can raise your hand or open your mic to discuss any issues with the workspace. If there's none, I believe we can continue. So first, we have interleave FASTQ to paired FASTQ. So here is the workflow or the WDL, which is the workflow description language, right here. It takes a single interleave R1 plus R2 FASTQ file. R1 and R2 here are the reads, so read 1 and read 2. So this is one file, an interleave FASTQ file. Then it will output separate read 1 and read 2 FASTQ files, or also called a paired FASTQ file. So the interleaved FASTQ file is actually a file that stores pair-end sequencing data, and the resulting paired FASTQ file will be an input for unmapped BAMs, which we will discuss in the next workflow. So here, maybe we can start by opening the data tab first to see which data we'll be using. So here in the data tab, we can see that there is a sample. And here in the sample table, there is one row, which is this NA12878 underscore small. And this will be the input of the example workflow we'll be working with. So here the entity type is sample, and we will be opening our workflow in the workflow tab. So we'll just click this. And since this is interleaved FASTQ to paired FASTQ, we'll be clicking on this workflow. So once that's clicked, you'll be seeing a page like this. And here, the data table is already set to sample. You can actually choose from a few here, but we're going to use sample. And then we'll be selecting the data. And here you can see that the row we just saw in the data tab appear here. So we'll just check that and we'll click OK. All right, so here in this page, you can actually see the script for running this workflow. And here you can just see the input files, which is the input FASTQ. Then here we can see the output file names, which will be the read1 and the read2. Here's the command for separating the interleaved FASTQ file. And these are the technical configurations, or like for the technical settings. And here the output file will be the read1 FASTQ and the read2 FASTQ. So this is the paired FASTQ file. And for the input itself, it will be the single file of the interleaved FASTQ file. And then after this, we'll just click Run Analysis. And then there will be a prompt where you may enter an optional comment, but we don't have to actually write anything. This is just for additional comments. So we'll just launch this workflow. So this will take a while to launch because here in the tutorial, it actually says around 3 minutes, but it actually varies depending on the file size or the resources available. But we'll just wait for the status to be successful. And maybe one thing I want to add is that this tutorial on the left, the link we sent, this is actually, it seems to be outdated. So the actual steps, for example, to run this tool on the sample file, we have to select process multiple workflows from sample from the tool menu. And if you can see in the actual page, we can't really find the tool menu. So we assume that the platform has been updated, but the tutorial hasn't been updated according to the features in the current platform. So maybe that's just one of we'll add. Oh, and there's a chat. Let's see. So we'll just refresh this page. And this is already succeeded. The status is succeeded. And to see the details, we can actually go here to the links column, and there is a job manager icon. We can just click on this, and it will redirect us to a new page. We'll just sign in. And here we can actually see the details of the job we just ran. So here is the input, So the input was the interleaved FastView file. And now we can see the output. We have two separate read1 and read2 FastView files, or the paired FastView. And here we can see other details. All right, so once we've done that, we can move on to the next workflow, which is from paired FastView. So we've just, the files we've generated or produced, the paired FastView will be converted into unmapped BAM. So here is a paired FastView will be converted to unmapped BAM formats and adds read group information. So like before, we just go back to our workflows and we click the workflow where it's paired FastView to unmapped BAM. And here we do the same thing. We select the data table. It's already defaulted in sample. And then we just select the data. Same thing, we check box. We check this row, click OK. And here the script is also given. We can actually see all the data being inputted, and then the commands. And the output file, which is the unwrapped BAM. So here we have quite a few inputs this time, and it's because this is a read group list. So the input there is a read group list in PSP format. For those of you who don't know, PSP format is tab space, I think. Tab space values. So it's like CSV, where CSV is comma separated values. So PSV is tab separated values. So it contains metadata and paths to Terra sequencing data in FASTQ format. So the metadata itself, there's read group, sample name, library name, platform unit, run date, platform name, and sequencing center. Here we can actually see the data, and all of this is available. If you scroll to the right, we can actually see the metadata over here. Okay, so we'll just go back to the tutorial, and then we'll just continue with the workflow, I guess. There's not much to discuss anymore about the background. So now we've seen the metadata, which is all here. And we'll just go and run the analysis. So same thing, there's going to be a prompt where you can add an optional comment. We'll just skip that. We'll click launch. And we'll just wait for the job to be successfully run. So maybe while we're waiting, we can just have a quick review on what an unmapped BAM is. An unmapped BAM file is actually it's a binary alignment map, where it's a file format used in bioinformatics for storing sequence alignment data. So this is a common file format that is used in genomic analysis toolkit, processing, and analysis tools. So we'll be working with unmapped them a lot. So I think this will take some time because the specification here is longer than the first workflow. It's actually 7 minutes. Oh, and it's already submitted. So I guess we'll just have to wait a bit more. We can actually open the job manager here. And we can see, oh, it's already succeeded. Let's try refreshing. Oh no, it's still submitted. We'll see the input. So this is the metadata we've discussed earlier, and the output should be an unmapped BAM. So I guess that's done. I'm not sure if the status here is succeeded or not. Let's see. Great. So now the status is succeeded, and we can move on to the third workflow, which will be presented by Katrina.
All right, thank you, Jacqueline. Okay, let's move on into the next step. The third one is theorem to BEM. Okay, so we need to convert from theorem format to BEM format. Let's see. Okay, the script converts file from theorem to SEM to BEM format. So we cannot directly convert theorem to BEM. Why? Because the reason this approach was chosen instead of converting theorem to BEM directly using SEM tools is because SEM tools 1.3 produced incorrect bins due to an old version of HTF-lib included in the package. SEM tools version 1.4 and 1.5 have an NM issue that causes them to not validate with Picard. So it produces incorrect bins and it caused them to not validate with Picard. So we cannot directly convert from CRM to BEM format. So we need to use this step, CRM first to SAM to BEM. So by the way, what is CRM? Okay, CRM is compressed read archive in SAM or BEM format. So using CRM, it is a compressed version of SAM or BEM file designed to reduce storage requirements while maintaining data integrity. So when we highlight the integrity process, then we need to reduce the storage. We can use this format. And what is BEM format? BEM is binary alignment map. DRAM and BEM are file formats used in bioinformatics to store sequence alignment data, typically produced by high throughput sequencing technologies like DNA. Okay, so the output will be BEM index, and validation report to a Google bucket. What is Google bucket? Here I use ChatGPT to explain that. So we have personal assistant that is smart. A Google bucket is a Google Cloud Storage bucket, commonly referred to as a Google bucket. It's a cloud-based storage container provided by GCP. Okay, basically Google bucket is Google Cloud Storage bucket. All right. So, like we already follow, like Jacqueline does, all we need is go to workflow. Then we find theorem to BEM workflow. We selected the data table sample here. Then we select the data, NA, blah, blah, blah. We can choose it. Okay, here is the script. We can see overview the script here. Workflow CRM to BAM flow. Okay, convert CRM to SAM to BAM and mix uh BAE, BAI. So like what I said, the output is BAM, the index and validation report. All right. We can go to the next, the input. CRM file, of course. Then we can click run analysis. It is optional to write it down in the describe box, but we can skip it. So click launch. Okay, the status is launching. We can wait until it is succeed. Okay, while we wait this process, we can go to the next step. Okay, there's a question from Agatha Silalaki. Why I can't click select data or run analysis? Is it necessary to clone first? Yes, yes. The first one, we need to go to workspace like Jacqueline show. If you guys already have sequence format conversion here, uh, yeah. If anybody who can clone, please make sure you guys have enough space. We believe that we only have 2 or 3 workspace. So if you have around 3 or 2 workflow, you can delete first like this. Delete, for example. Then we type delete workspace. Click delete workspace. So, Agatha, have you find this workflow sequence format conversion?
Tadi udah nyoba mau nge-clone yang sequence format ini, tapi karena ada kendala itu, Kak. Jadi lagi ngehapus apa gimana ya?
Yes, yes, yes. How many workflow do we have here in workspace?
Masih yang kemarin itu, yang Terra data tables, tapi ini mau aku hapus dulu, Kak.
Yes,
Biar bisa
okay.
sequence-nya.
Good. Okay, thank you, Kak Agatha. All right. If you have another error or issue, please share to chat column. So maybe we can share what we know. Like this case, if you cannot clone the workflow of sequence format conversion, we need to have enough space. Maybe we can delete the previous workflow first. So we can clone this workflow and we can do some repurchase the workflow. Okay, maybe let me continue the process. Okay, the status from launching move to running. Let me refresh. Okay, it's still running. So the based on this article, it said that we need 7 minutes to run this process, and it costs $0.02. Okay. Next, we want to convert BAM to unmapped BAM. Okay, this tool is important because unmapped BAM files are the file format generally accepted by most GATK analysis workflow. So, if you guys have any question about why do we need to convert to unmapped BAM, the answer is this format file generally accepted by most GATK analysis workflow. Okay, there is chat from Kak Agatha. He has already cloned the workflow. Okay, good. Okay, this tool accepts single or multiple BAM files. It is configured to accept data from a table in the data tab. An example BAM file is added to the sample table. Okay, so we can use the sample data from the Terra Bio, or we can also use our own data using the SV format to load. Then we can load the file. It needs 1 hour and 3 minutes, I guess, and it costs $0.06. So let's get into it. Go to workflow, then we choose BAM to unmap BAM. That is unmap BAM files that is generally accepted to GATK. We can review the script here, overview the script here. The input is BAM file, of course. Okay. The output is unmapped BAM. There's task sort SAM, and here's the command. Here's the runtime. Yes, the output is sorted BAM. Okay. BAM file as input. So we can select the data table as sample, and we can select the data by check this one, click OK. Then we can run analysis. You can fill it, or you can keep it blank. Click launch. Then after that, we can get the unmapped BAM format file. So that is the process of converting from CRAM to BAM and BAM to unmapped BAM. So guys, if you have case want to convert the format, want to convert file format to from CRAM to BAM or BAM to unmapped BAM, you can use this workflow, so it will make your job easier. That's it from me. Next will be continued by Rashidan. Please, Rashidan. There's still no voice, Rashidan.
There is voice.
Okay.
Yeah, okay. Okay, so the next one is validate BAM. BAM is stand for binary alignment map. So this workflow performs format validation on sequence align map or binary align map files in a list. So the input requirement is one or more SEM or BEM files to validate, and explicit request of either summary or verbose mode in inputs. So the outputs will be a set of text files, or we call it notebook, notepad or notebook, and no, notepad files containing the validation reports, one per input file. So the sample data that we will be use is NA 12878, like before, with the sample total size is 5 gigabytes. So let's go forward for workflows. So we choose the validate BAM, and here is the script of validation workflow. So here's the input function. So the input is input BAM, and there is like a summary or verbose one. Okay, here is the input file. So there is a input BAM and the string output for the text file, and in the validation mode. So yeah, this is the script for validate validation, and the input is here. So it only need one input, as the variable is BAM array. So the type is array, and the output will be a validation report in TXT format, or maybe notepad files. So we could just run the version below this one. Yes, so we can launch this. So yeah, it's already run, and we should maybe waiting around 19 minutes maybe. Yes, that's it. Okay, so we go for the next one, which is validate variants after we validate the BAM files. So this workflow perform format validation on some or BAM file in list 2, but the input requirement will be one VCF file or a key VCF file and its index, and a list of interval to process for parallelization, and the genomic resource for reference genome, and the format will be FASTA, or there is like FASTA FA or dictionary. And the output will be same as before. It's TXT file, but it's more like a lot of lists. So a list of text files for each scatter in chunks containing the tool standard output, which will contain the relevant error message if the tool encounters a validation error. So if there is a validation error in the tools or maybe the data, there will be like error message in the output too. So the sample data is same as before. So we use NA 1287881 with the time and the cost is same as before. So let's try the workflow. So yeah, we use a different script too. So the difference with the input from validate PEM and validate variance is for validating variance, there are like more file, which is VCF and VCF index. While the validate PEM1 is there is no need to input index, but and here we should input the variant index in here. So also it's a list of interval to process. Well, this is there is no a list of intervals, because in this script we use parallelization to validating data. Yes, and the file format should be a FASTA or FASTA index or dictionary. So here it is. So the output is same as a validate BEM, so it will be validation report, but it's more like more likely there is a list of text files, so there is a lot than validating BEM1. Also there is like a error message in the output. Yeah, so there is input definition in here from the script, which is input variance and variance index, interval list, dictionary, and FASTA file. And the output will be validation report, same as BAM validation. So we run the analysis. Okay, so it's maybe we should quite in here the status. We can see the status in job history. So the validated BAM1 is already done, and we will waiting for a validated variance one. And we could maybe check the notepad file or txt file, or maybe we call it maybe a validation report in here. So because this entire workflow need a computational power from a Google Cloud platform, like this is need $1.13. We could check it from here. So this is a Google Cloud platform. You can use this one, maybe I will copy in here. So you can see your bills in here. So this entire workflow we use, it costs around almost $1. So this is it. You can see the report here too. So we are given $300 from Google Cloud. So this around this, and we already use around maybe like around $2 to $3. So yeah, you can see the entire cost or billing of Terra from this Google Cloud console. Yeah, so there is overview and the reports of bills. So you can calculate your billings from Terra Cloud computation. So maybe that's it from me.
All right, thank you, Rashidan. Okay, guys, is there any issue or questions from you all? Okay, let me review what we already discussed. Okay, there are so many format files in genomic field, and every format has its own function. There are CRM that used to um so we can have file that is compressed, and we have unmapped them that generally accepted by GATK, and we have PCF file that can be analyzed. So every format has its own function. Then if we have um, needs to convert from one format to another, we can use this workflow. We can reproduce or reuse this workflow. So we are not working from scratch. That's our material today. Hopefully this um, knowledge can be useful for us in the next day. Okay? If there is no question, yes, please. Is there any question? Okay, thank you. Okay, let let me close this session. Thank you all for the coming and for following this tutorial. Hopefully we can meet again in the next time, inshallah. Thank you all and see you next time.
Okay, Kak. Thank you.
See you.